Assalamu alaikum guys. My name is Dr. Omar Khan and I am assistant professor in the Department of Botany at Pakistan Shipowners Government College. Today we are discussing the second reaction of the photosynthesis. The photosynthesis is the manufacturing of the food material with the help of carbon dioxide and water, and oxygen is released as a byproduct inside the chlorophyll molecule. So there are two different types of reactions in this photosynthesis. One is the light reaction and another one is a dark reaction. It's actually called the light dependent reaction and the dark light independent reaction. So today we are discussing the dark reaction of photosynthesis, which is present in the chapter 11, the bioenergetics, and page number of 254 in the second it's not the latest version of the syntax book, but it's previous version. Now in the latest version it may be in different page. So the dark reaction of the photosynthesis is actually it's a chapter of the first year botany, and that's my introduction. Now what actually the dark reaction is also known as the Kelvin-Penson cycle because of the scientist name, C3 cycle, the light independent reaction, and the fixation of carbon dioxide. It's the most important name. That's a fixation of carbon dioxide because anyhow the plant is going to fix the carbon dioxide for the manufacturing of food material. So the site of the reaction is where the reaction is to be occur is the stroma of the chlorophyll molecule. Chlorophyll molecule we have two different types, especially present in a chlorophyll, the grana and the stroma. The grana containing the light reaction, the light dependent reaction, and the stroma is containing the dark reaction. So stroma is a liquid gel present inside the chlorophyll molecule. Now the first stable compound during this C3 cycle is the 3-carbon compound. That's why it's called the C3 cycle, and the phosphoglyceric acid PGA is the compound which is the first stable compound formed during the dark reaction. What actually the starting with the ribulose 1,5-biphosphate is a compound 5-carbon compound present in every molecule of chlorophyll in every plants. It's actually deal with the carboxylase, the ribulose 1,5-biphosphate carboxylase is an enzyme which is necessary to fix the carbon dioxide. It's a 5-carbon compound and it's a enzyme which act on this 5-carbon compound. They have 3 different steps in this C3 cycle. The one is the carboxylation, which is actually the fixation of carbon dioxide, and the reduction and the regeneration. The 3 different steps are there. Now the products which is formed during the C3 cycle is actually the glucose molecule, which is the most important. It's actually the 6-carbon compound, the glucose C6H12O6. The consumption, what actually the utilization of the chemical energy during this dark reaction, as well as the different carbon dioxide which is from the atmosphere and inside the stroma. So that it's a light due to chemical energy which is produced. Where it is produced inside the light dependent reaction. It's a light reaction. Now what actually the process is, we define it in the 3 different stages. One is the carboxylation, and then one is the reduction, the third one is the regeneration. Now this carboxylation is in this step the addition of carbon dioxide with 5-carbon compound, it is ribulose 1,5-biphosphate, which we discussed in previous slide, to form the 2 molecules of 3PG. It's actually forming the 5-carbon plus 1 carbon dioxide carbon. This is called the 6-carbon compound. That this 6-carbon compound is formed in 2 different molecules, the 2 molecules of 3-carbon. The 3-carbon compound is 1 molecule, and 3-carbon compound is the molecule which is 6-carbon compound. The first stable compound is the phosphoglyceric acid, which is formed during this carboxylation. Now the reduction. By utilization of the chemical energy, the 1 molecule of PGA, phosphoglyceric acid, form the glyceraldehyde phosphate. It's isomer of PGA, and the other molecule is molecule from dihydroacetone phosphate. It's also another 3-carbon compound which is isomer of PGA. Now the one ribulose 1,5-biphosphate combined with another carbon dioxide form the 6-carbon, which is split into a 2 molecules of 3 different carbons, and they form the gap and dap. Now in the regeneration, it's a regeneration, the one molecule of 3 carbon compound utilize its 3 carbon and another 3 carbon compound utilize 2 carbon to form. It's actually the reformation of this ribulose 1,5-biphosphate, and the 1 carbon is remain, and it's actually we get the 1 carbon from the, which is from carbon dioxide. Now what we deal with this 1 carbon compound, it's actually we see in the process in the cycle. Now it's a blurring slide, which actually it's so easy to understand because we have a different cycles in our book. We have a circular cycle and different types of books representing this cycle into a different ways. But in my opinion, this slide is so easy to understand in first year class, so I can explain it. If I explain this slide, then you can easily understand this. Now this is the ribulose 1,5-biphosphate. It's a 5 carbon compound. You can write it down with your own handwriting. That's 6 is a 5 carbon compound which is present inside the chlorophyll molecule. Now this 5 carbon compound attached with this is the carbon dioxide, and that is the enzyme. Now we form with this have 6 carbon. Now this is the water molecule, is form the phosphoglyceric acid. Now this is phosphoglyceric acid. The whole 3 molecules of ribulose 1,5-biphosphate, ribulose 1,5-biphosphates combine with carbon dioxide, and then in the water molecule, and that is the PGA, phosphoglyceric acid, and the phosphoglyceric acid. Now during this formation of phosphoglyceric acid, the chemical energy which is utilized, it's the 6 ATP and the 6 NADPH. That means the 1 ribulose 1,5-phosphate when converted into a PGA, that is the 2 molecules of ATP and 2 molecules of NADPH. So when we have a 3 ribulose 1,5-biphosphate, we have a 6 ATPs. We utilizes the 6 ATPs and 6 NADPH. Now that is actually the consumption of the chemical energy during this carboxylation. Now that PGA is converted into the gap and DAP, and the last one is the gap. Now what actually, this is the isomer of the PGA, the gap, glyceraldehyde phosphate, dihydroacetone phosphate. Now the glyceraldehyde phosphate, the dihydroacetone phosphate, the glyceraldehyde phosphate, the dihydroacetone phosphate. What actually happening after that formation of these molecules, they again regenerate into this ribulose 1,5-biphosphate. How it is happening? The 3 molecules, 3 carbon compound, and also there is this actually utilizing all their 3 molecules of carbon and 2 molecules of from DAP. Okay? Now that actually forming the 5 carbon compound. This 5 carbon compound forming the ribulose 1,5-biphosphates. We remain 1 carbon from that gap because it's a 3 carbon, 2 is utilizing to form the ribulose 1,5-biphosphate, remaining 1 carbon. This 1 carbon is combined with this gap. So this is the 3 carbon compound attached with the 1 carbon, 4 carbon. They spare 1 carbon from that DAP, dihydroacetone phosphate. So it's actually forming the 5 carbon compound. 3 from the gap, 1 from DAP, and 1 from their left one DAP. So it's actually forming the ribulose 1,5-biphosphate. It's a 5 carbon compound. Now again regeneration of the ribulose 1,5-biphosphate, and the third one where we get the 2 molecules of carbon here is sphere and the 3 molecules of carbon gap. That's forming the 5 carbon compound. We again regenerate the ribulose 1,5-biphosphate, ribulose 1,5-biphosphate, ribulose 1,5-biphosphate. During this formation of the ribulose 1,5-biphosphate, we also the plant is utilizing the 3 molecules of ATP again, and they form the ADP. It's actually reduced. Now, what we actually getting that this gap is remain where it is realizing. It's a 3 carbon compound. It's 3 carbon compound, and what we need to form the glucose is the 6 carbon compound. So what we need to do, what the plant need to do to form the 6 carbon compound hexose sugar is actually repeat this whole cycle into another time. So if after repeating this cycle, the gap, another gap is formed, the 3 carbon compound, another 3 carbon compound formed, actually forming the 6 carbon compound. Now this 6 carbon compound is forming the glucose. Now that means the C3 cycle, when the C3 cycle going twice, then the sugar molecule is formed. And when the C3 cycle going twice, that means the double energy is utilizing. In a single cycle, the 6 ATP and 6 NADPH is utilizing, and there is the 0. So 9 ATP and 6 NADPH in a single cycle. So it's actually the C3 cycle of every plant to form the glucose molecule. So it's a process, it's so simple as compared to the circular cycles. Now what actually the energy balance in this cycle? The total energy utilization of is 9 ATP and the 6 NADP, as we discussed in the previous slide, for the 3 carbon compound. Now for 6 carbon excuse me, the 6 carbon is a 3 carbon के लिए 9 ATP and 6 NADPH. Now the 6 carbon compound, the 18 ATPs and 12 NADPH. As we discussed, the 6 carbon containing the required 18 ATP molecule and the 12 NADPH, and for 1 carbon, as we calculated, the 3 ATP molecule required and the 2 NADPH molecule required during one light dependent reaction. What actually the light reaction we get, because this ATP and NADPH is actually utilizing the energy which is produced during the light reaction. Now what we get during the light reaction, because we need the 18 ATP molecules and 12 NADPH molecules for 1 glucose molecule. So what actually it's a NADPH, 1 NADPH is equal to the 3 ATP molecule. If we calculate the total amount of ATP, direct or indirect, is different. If we discuss the kilocalories, 1 NADPH molecule is equal to the 52 kilocalorie, and 1 ATP molecule is 7.5 kilocalorie. So we calculate it in our respiratory cycles. During because it's actually the respiratory cycle is a energy producing cycle. During one light reaction, light dependent reaction, we get 2 ATP and the 1 NADPH. That means we get a very little amount of the chemical energy during the light reaction, and we need a huge amount. It's at 18 ATP required or 12 NADPH, and we only get the 1 NADPH during the one light reaction. So how many light dependent reaction required for one dark reaction? It's a question of how many light dependent reaction is required for one dark reaction. We easily calculate it. And how many light reaction for 1 glucose molecule? It's a one dark reaction is actually the 3 carbon compound. It's not a glucose molecule. So how many light reaction required for glucose molecule? It's easy to understand. So how many light dependent reaction required for 1 dark reaction? That means the 3 carbon compound. How many light reaction required for 1 molecules of 3 carbon compound? It's a 1 dark reaction, C3 cycle, one cycle of C3 cycle. Another question is, maybe it's occur in this form, that maybe how many light reaction required for 1 6 carbon compound? It's a glucose. So that is the question, and you can easily calculate with this equation. So we actually go through our review in this dark reaction. What actually we studied, the dark reaction, the C3 cycle, and we know that every plant which is the autotroph and this manufacture their own food is definitely go through this dark reaction or C3 cycle. Ultimately they have to prepare their own food because they need energy. So the site of the reaction is the stroma, and the first table compound is the PGA, And now this is the starting product, is the start where the reaction is to be started is ribulose 1,5 biphosphate, a 5 carbon compound, and the enzyme is ribulose 1,5 biphosphate carboxylase, which fix the carbon dioxide. The 3 steps: carboxylation, reduction, and regeneration. Now the product is formation of the glucose molecule, which is the most needed molecule for the energy purpose, which is breakdown during the Krebs cycle and release the energy, glycolysis in Krebs cycle and release energy. We study in our next classes or next slides. Consumption of the energy is the chemical energy which is produced during the light. It's the most important reaction. That's the light dependent reaction, is actually the conversion of the light energy into a chemical energy. How it is converted into a light energy into a chemical energy, it's a different question. It's a different cycle. We study in our previous light reaction already. So the process is 3 different steps. One is the fixation of the carbon dioxide to form the first stable compound, and then converted into their isomers, gaps and dap, gaps and daps, gaps and gaps, and then the 3 molecules of carbon regeneration is occurs. A 5 ribulose 1,5 biphosphate is regenerated, and the remaining carbons are from the 3 carbon compound gap. It's a simple cycle. You can draw it on your because this cycle is not as a common. It's I got from my teachers, so that is the most simple to understand the dark reaction. So the carboxylation, there's reduction, and then the regeneration. So we get easy understanding from this. So the energy balance is it's a during the 3 carbon compound, it's a 9 ATP and 6 NADPH, and then if we have a 6 carbon compound, then we require 18 ATPs and then 12 NADPH. So during the one light reaction we get only 2 ATPs and 1 NADPH. So how many light dependent reactions required for one dark reaction, or how many energy is required for formation of one glucose molecule? So thank you very much, and definitely during these timings you understand these types of lectures and helpful for your classes, next classes, and we are keep in touch with this presentation with these types of lectures. Thank you very much. Love.